this is the basic session that I'm going to cover, like what we basically do in the single cell. So from that part you can read, and then you can start your analysis part based on the resources I'm going to provide. So you can read the stuffs and whatever you want to do. For example, if you're going to do any kind of projects, or if you're going to learn these kind of domain, so you can have some basic knowledge, and then you can start reading more stuffs, more papers, and you can do and analyzing any kind of data. You can collect it from any papers. So this entire session will be the basic of single cell, and like, so what is the reason of shifting to single cell, and what is the advantage over the bulk RNA-seq. So we are going to cover everything. So starting with the first part. So before single cell, uh, I guess like you may aware of the NGS workflow, like from where sequencing data and high throughput data is coming from. Okay. So the entire workflow is divided into four domain or four group. So first is basically the extraction part, means you have the samples and you are going to isolate your genomics, or you can say your DNA or your RNA. So you need to extract those information, genetic information from the sample, and these information totally depends on the experimental approach or the analysis part you're going to do. Let's say for some example, someone is going for the variant calling, or let's say for the mutation calling, so they're going to extract the DNA part. Let's say someone is going for the transcriptomics profile, so they can collect the information on the transcript. So this is the first step for the entire NGS workflow, that is basically dealing with your sample, extracting the genetic material. Second step is basically the library preparation. So library preparation is needed because we have to prepare some reads that is going to, we have to send it for sequencing. So there will be a sequencer machine, that is the part 3. So all has their different capacity and capabilities. Okay, so we have to provide the reads that should fit the condition. Let's say for example, if I have a sequencer, let's say I have one sequencer, and this sequencer can do the sequencing of reads, that is basically the length should be around till like less than 150 base pair. Means if there is more than 150 base pair of read, we can't do sequencing of those particular reads. So this is the condition. So what we have to do in the library preparation, the DNA we are extracting from the samples, so that is a big stretch of DNA. Okay, so this is more than 150 base pair, this is more than 150 base pair, let's say for example. So this is not fulfilling the condition for the sequencer, means the sequencer requirement is less than 150 base pair. So the length of the read should be 150 base pair. This length should be 150 or less than 150, let's say for one sequencer machine. So what we have to do, we have to do the fragmentation, means a bigger template or big stretch of DNA we need to fragment into fragmented DNA. Okay, so this is a part of fragmentation. And we can also say, for example, when we are dealing with, let's say, the metagenomics data, or we will have the short reads, basically 16S RNA sequencing, so that time we call amplicon. Okay, so those are different concepts you can read about. So let's say for example, overall the basic concept is we will have a bigger stretch of DNA, and we are going to do this fragmentation to create a fragmented DNA or small stretch of DNA. Okay, now this stretch of DNA will be less than 150 base pairs. Now it is going to fit the requirement, that is for the sequencer. Let's say some sequencer may have like the potential to do the sequencing for 250 base pair, 350 base pair. So different sequencer has different capacity to do the sequencing part. So some sequencer is for the short reads, some sequencer is for the long reads. For example, Illumina is for the short reads. Then this is Nanopore is for the long reads, where we provide the long stretch of DNA for the sequencing thing. Okay, so this is the first concept. After extraction, we are going for the library preparation, and library preparation is basically doing the fragmentation, adding some of the things to the reads. So the fragmented DNA or the DNA we are going to send it for sequencing. In biological term, in the NGS workflow, we call them as a read. Okay, this is a read, one read. So at the end of the read, we are going to add couple of tags. We will discuss what is the usefulness or importance of these tags in the upcoming slides. But as of now, just get to know this is green part is your reads or fragmented DNA, and we are going to add the adapters, barcode, and a lot of information. So those you can consider as a tag part. So as of now, we have the fragmented DNA and we have the tag associated with the fragmented DNA. And these things we are going to send it for the sequencing. So the third step is sequencing. We are providing the reads that need to be sequenced. Means we want to know the sequence, what exactly the sequence is present on this particular, what nucleotide base is present, whether it is ATGCCC. So what nucleotide base present on these fragmented DNA, I want to know. So to know those things, I have to send my sample for sequencing. So this is the third part, that is sequencing. And after sequencing, as you know, from the sequencing part, the output is basically the raw data, that is the FASTQ file. So FASTQ file is nothing but only has the sequence and the quality of the sequence. Means, as we say, for example, this is read 1, read 2, read 3, read 4. Let's say we have 4 reads. So in the FASTQ file, what information you will have? You will have 4 reads and you will have the sequence. This is the sequence like AAC, GTC. So this is the sequence of one read, and also we will have the quality of each nucleotide base. Okay, if you are aware of the corporate quality, so those things basically the concept of quality score of each nucleotide base. So I hope even if you have watched my YouTube videos on the genomics file format where I have covered the different biological files format like FASTQ, FASTA, VCF, SAMBAM. So in FASTA and FASTQ, I have covered what is the sequence and what is the Phred quality score for each nucleotide base. If you're not aware of that, you can cover that part using my YouTube video channels. Okay. So this is the fourth part. So starting with the raw data that is coming from the sequencer, and this raw data basically consists of the sequence and the quality of the sequence. Okay. And then now what we have, let's say, as of now still we can't have all the information. Means as of now we only know the reads, okay, and the sequence of the reads. We are not aware, okay, these reads are present on which genomic region in my chromosome, or let's say your genomic region, you have the entire genomic region. So these reads are going to map at which regions. As of now we are not aware of that. So for that we do the alignment. So from FASTQ to BAM conversion is basically the part of alignment. Alignment in the sense you will have the reference, for example, from wherever you have taken the sample. Let's say this was a sample, let's say either human, mouse, whatever the sample or organism you are using to collect the sample. So you need to take the reference of that part from any database. So let's say I'm working on human sample, let's say human tissue. So I'll take a reference of human. So I know the entire sequence or entire nucleotide base of the entire genome of human. Let's say this is the reference. So this is your reference. And whatever the reads we have that we have collected and fragmented, so we have a many reads. Now what we have to do, we have to map, means we are going to map the reads against the reference genome. So wherever they are mapping or finding the complementary thing, they are going to map it. Okay. So when we have the mapping, so this process is called alignment, means we are mapping our reads against the reference genome. So this is the alignment. So this is the way to find the sequence of your query genome. So this is the reference genome. This is your query genome. But as of now, your query genome is split into multiple short reads. So what you're trying to do, you're trying to map, and after mapping, you can overlap the sequence and try to build a stretch of DNA. So this will be your queried sequence, or this will be your reference sequence. And after mapping, you will get to know the entire nucleotide sequence of your queried DNA or your queried genome. Okay, so this is the part of alignment. And after having those information in the BAM file, we can go on different mode of analysis. Let's say someone is going to study the, let's say for example, variant calling, germline, somatic caller. So they are going to extract the DNA and they are going from the BAM to the VCF file conversion, means going for the variant calling and other kind of method for generating the mutation or list of files where we have all the mutation. So that is basically stored in the VCF file. Okay. But for the single cell, we are dealing with a transcriptomics profile. Transcriptomics means the transcript profile, or you can say the gene expression profile. Okay, so we are not going for the variant calling. From the BAM file, what we will do, the information we have extracted is basically the mRNA, and after having the alignment, we have the BAM file, and from the BAM file, we are going to get the count matrix. Count matrix in the sense the number of transcript for each gene that is present within the sample. So this is the count part. So that from the BAM file, we can have the feature count function to generate a count matrix. Okay, so this is the final outcomes. So our goal is do the all the steps, and after alignment and everything, we should have the count matrix. So this is the entire workflow for the NGS thing. Yeah, so starting with like how we can collect the gene expression data. So before the single cell, there were a lot of experiment people used to generate the gene expression data in the form of microarray, and the second one is the RNA-seq. So if I go quickly on explaining the part of microarray, so it's very simple. So in microarray, basically we are checking or we are comparing the gene expression profile between two conditions, whether it can be healthy versus pathogenic cells or tumor versus non-tumor cells, anything, like it can be two conditions. So what we are trying to say is we are collecting the RNA. So we have the cell types. Let's say, for example, this is the healthy cell culture. This is the pathological cell culture. We have the culture. From culture, we isolated the RNA, and then we go for the reverse transcription and we create the complementary DNA, let's say. Okay, so now also we are adding a tag. So each fragment of DNA, let's say, or you can say the RNA, will have a tag. Okay, so we are going to add a tag. So one tag will be there for the healthy sample, one tag will be there for the infected sample. Okay, so let's say I'm going to add a green fluorescent tag for the healthy part and red fluorescent for the pathological part or the infected part. Now, what is the principle or backend of the DNA microarray is basically we will have the chip, and within the chip we will have a short, you can say, short circle well, and this microarray is basically the probe based, means whatever the information we want to collect, we are going to design the probe for only. Let's say, for example, I need to check the expression profile of 5 genes, so I'm going to prepare the probe for those 5 genes so I can capture. My target is to capture or isolate those 5 genes expression profile only. I'm not worried about whatever the information or transcript that is coming out from the cell. I'm interested to look for the expression profile of only the 5 gene. So what I'll do, I'll create a probe that will bind, let's say for example, from this sample there is a gene A transcript. Okay, so what I'll do in this particular plate, in one particular circle, I'm going to add a probe. This probe will be the complementary of this particular gene A transcript. Okay, so let's say this is a probe I have created. Okay, and let's say in this particular cell, let's say we are going to, let's say this is a particular cell where we have to add a couple of things. One thing is going to add a probe that will be specific for gene A. So there will be gene A, and we are designing a probe that we need to add that will bind complementary to this particular transcript that is for gene A. Okay, and we have a sample, like we have collected all the transcript within this particular test tube, let's say, and we have a specific transcript that is for the gene A. So if you're going to add everything to this particular dot, let's say, so there is only one thing that is complementary for the gene A. The rest of the thing will not bind or do complementary sequence. There is only one transcript that is going to do the complementary with the gene A probe. So whenever this part that is coming from this sample and this part that we have designed the probe thing that will do complementary to each other. So whenever they form the bonds, then we will get the fluorescent signals. Means we have added a tag. When there will be binding, then we will get the signal. So it means this particular gene that we were looking is present within the sample. Okay. If we are not getting any signal, it means there is no expression of this particular gene. Okay. So this concept is clear. So same thing we are looking for, let's say for example, in this particular thing, what we did, we collected all the transcript from this sample, put it on this particular dot, and I have added a probe for, let's say, gene A. Then if you get a no signal means gray color, let's say in this particular example, it means neither gene A is present in healthy cell, neither it is present in infected cell. If you are getting a red signal, it means it is present in the gene. Let's say in this particular thing, I added a probe for gene B. Then I got a signal of red. It means the expression profile for gene B is present within the infected sample, but it is absent in the healthy sample. Okay. Same thing if you are getting a signal of yellow, what it means? It is present in both the condition. The gene D we have added in this particular thing, for example. So we can see the gene D is present and showing expression in healthy cell also and in infected cell also. And if you are getting a green, it means the particular gene is showing expression in healthy condition only, and it is absent in the pathological cell or infected cell. So this is the concept of the microarray. So this is the one thing. So but there are limitation. So limitation is because we are just measuring the transcript that is based on probe. Means we can target 5 transcript, 10 transcript, let's say, but we can't capture the novel transcript or the rare transcript or some new transcript, okay, or some finding new variants. So these things we can't do with microarray. So that's why there is a new technology that comes after microarrays, basically RNA-seq. So in RNA-seq, this is not dependent or rely on any pre-designed probes. Okay, I'm going to capture 10 transcript. I'm going to check for the expression profile of 10 genes, 20 genes, 50 genes. This is not the way. I'm going to extract all the genes from the sample. So it will be like all the RNA molecules present within the samples. I'm going to check the expression profile. So this is the concept of RNA-seq. So this is the difference of microarray and the RNA-seq. Okay. Now after RNA, we shifted to a new technology that is single cell, and the recent technology that we are developing or we are working in the field, that is the spatial transcriptomics. All are the analysis of transcriptomics, mean gene expression data, but technology is different. There are some of the pros and cons associated with each technology. So let's try to understand what exactly the meaning of single cell and how it is different from RNA-seq, that is bulk RNA-seq, and how it is different from spatial also. So let's say bulk RNA is basically the RNA-seq analysis part. So in this particular thing, what we basically do, let's say with an example I'm trying to explain. Let's say if I'm making a fruit juice, okay. So in bulk RNA, we are going to add a couple of fruits. Let's say I am going to add 2 apple, let's say 1 banana, 2 avocado, and 2 strawberry, let's say. So whatever the fruit content that I'm going to add, that will give the flavor of the taste of the juice, okay. And if I'm going to ask you the same thing, could you please let me know, or you can give me the answer like how many avocado I have added, how many strawberry I have added. So you can't provide the solution because you're not able to provide the solution because now we are mixing everything. So we are losing the information of the content side. How many things I have added to prepare this juice. Okay, so in bulk RNA-seq, what is happening? We have the entire tissue and we are collecting or isolating all the RNA molecules into, let's say, in a pool, but we are losing the information this RNA is coming from which cell. Okay, so we are losing the information, we are just having all the transcript. So we can say this is giving the average expression profile, because if you are aware of that, we have a unit of life, that is cell, and when you are going to combine multiple cells, it is going to create a tissue. So tissue is basically made up of multiple cells. Okay, so let's say a single tissue can have different cells. Let's say this is a tissue which consists of 5 cells. Okay, so gene A can be present in this particular cell also, can be present on this particular cell also, can be present on this particular cell also, and let's say this is absent in these 2 particular cells. But when you're doing for bulk RNA, you will not get to know. So let's say you are checking the expression profile of gene A. So whatever the value you are getting, that is going to represent the average thing. Like means whatever the value you are getting, that will say the particular gene is showing expression on the entire tissue. But that is not the reality, because the gene was present only on these 3 cells. But these 2 cells do not showing the expression for that particular gene. Okay. So these things were missing in bulk RNA. So those problem were sorted out in the single cell profile. So single cell profile means we are doing the same thing. We are doing the RNA-seq, but at single cell level. Means what we are doing in bulk RNA, we are rupturing the tissue and collecting all the RNA thing. But in single cell, we have the tissue. First thing we have to do is isolate single cell. Means let's say this particular tissue is consist of, let's say, 5 cells. So I'm going to isolate all the 5 cells first. Then I'm going to isolate all the transcript mRNA from each cell. Okay. So now I'm capturing the this RNA information, or you can say gene expression profile, at a single cell level. And this one was at tissue level. So this one was giving average expression profile, and this one was giving me expression profile at a single level. Okay. Now let's say this is a single cell part. So this is one advantage, because now I can say how many things you are going to add to prepare the things, because we have the quantity information, like from where the quantity is coming. For this part, we are not aware of exactly the quantity is coming from which cell, but now I can say the quantity is coming from which cell. This part is for the single cell. But one thing we are losing at single cell is the location. Means let's say, for example, if we have a brain tissue, so what we are trying to say, let's say we just isolate a region. Let's say I want to capture this tissue at this region. So we have the tissue and we isolate the cell, but we are losing the location. Means whether the cell is located at this region or at this region or at this region. We are not keeping these information in single cell. Means we can't say my particular cell is present exactly at which location. So these things is missing in single cell. But in the recent technology, that is the spatial transcriptomics, where we are doing the single cell plus we are capturing the spatial information. Means I can say exactly my cell is located at which position. So you can see the structure, how the fruit is organized. Same way, we will get to know exactly my cell is located at which regions. You can say at the periphery, we have the strawberry layer, then we have the grapes layer, then we have the orange. Same information we will have, like where we have the cells for the hindbrain region, midbrain region. These information we can collect from the spatial transcriptomics technology. That part was missing in single cell. But in today's lecture, we are going to cover the single cell, like we can have the gene expression profile at a single cell resolution. Yeah. So now this is the difference between bulk and single cell RNA. So what is the advantage and disadvantage? So because in bulk C, yeah. So in bulk C, the disadvantage is we are losing the heterogeneity part. Heterogeneity part in the sense the cell or tissue is consist of many cells, and you can say many different cells community. I mean, let's say for example, we have a cell. Let's say this is for the, let's say, brain data or something. So it can consist of different cells. It can have the granular cells. It can have the aldox cells. So multiple cell types, or you can say community or different cells that can consist of, that can be the part of this particular tissue. So we are losing the heterogeneity. So means we are rupturing the entire tissue and we are not keeping those information. We are just extracting all the transcript information from the tissue. So we are losing the heterogeneity part in the bulk RNA. And this is bias due to dominant cell types. What it means, for example, same way I was saying, I was preparing a juice for you, let's say. So I have added, let's say, 3 orange, or let's say one banana and one piece of apple. If I'm going to ask you, so there might be chance you're going to say the juice is made up of only the orange part, because there will be less taste or no taste for the apple part, and let's say the half banana part, because the quantity is less. So that may be case because there will be bias, because whatever the things that is present in high quantity, that is taking in charge to give the context, like the juice is made up of. So this can be the bias thing. So yeah, so this is the disadvantage, like there may be bias due to present of some dominant cell types. Okay, same thing that I was explaining with the juice example. But there are a lot of advantages, because if you have to simply just compare between two samples based on condition, like you're doing the drug treatment, you're doing some other condition treatment, so it's a very cost effective. Like you have to spend, low cost to do the analysis part and generate and do the comparative analysis, and there will be less technical noise. But when you go for the single cell, it is costly. Also, it is a complex process for the analysis part. When you see the analysis approach, that is a little bit complex, but in the bulk RNA, that is straightforward to do the comparison to find the genes that is showing expression in one condition and not showing expression in other condition. Okay, but there are a lot of advantage is cellular resolution, means you can have the expression profile of all the genes at single cell resolution. This is the first advantage. Second advantage, we can find the tissue heterogeneity. It means like we can get to know whether if we are working on lung data, breast cancer, you can say brain cancer data. So we'll get to know my tissue consists of which cell types. Okay. We can also find if there is a rare cell type that is not explored as far. Okay. So we can also find the rare cell types. We can study the trajectory of the cells. Trajectory of the cells means basically when you're going for the development biology, means you're going to study the development, how the development is happening. Let's say you're going to study the embryo development, or let's say you're going to study the plant tissue development. So where it's not like that. For example, if I'm going to say you have some cell types, so that those all cell types should not be present at some baby thing. Okay, so because initially we have a few cell types and some of the cell type is going to mature into different cell types. Okay, so that is a part of evolution, or you can say development. So those things we can also study with the help of trajectory analysis in the single cell. So these are the few advantages of single cell over the bulk RNA. And also bulk RNA has some advantage which include the costing, and simply we have to do the comparative analysis between two samples. So we can simply go for bulk RNA. No need to go for the single cell unless you need to find the cellular heterogeneity, study of development thing, or finding the rare cell types, checking the expression profile. These things, if you have the requirement, then you can go for the single cell study. Now, there are many applications of single cell study. So I have listed some few. Let's say finding the cell type means the heterogeneity of the tissue. The entire tissue consists of which kind of cells. Okay, let's say for example, if I'm going to take a blood sample, so blood sample will not have only the RNA, like the this one RBC. It can have the T cells, B cells, monocyte, macrophage, a lot of different kind of community of the cells within the blood sample. So basically, I'm going to find those community. So that is a part of cell type identification. Means the entire sample and the entire tissue is consist of which kind of cell types. Okay. Then the second one is a development biology. Development biology in the sense we are going to study sub-development change, let's say embryo development and other kind of development also. So for cell type identification, this one is the example. Let's say we are going to study, for example, lung data. So lung data may consist of different kind of tissue. So entire tissue consists of different kind of cells. So to find exactly what are the cells community that is present within this particular tissue samples, so we can find those things using single cell. Coming back to development bio, let's say someone wants to study the development cycle of any fish. So we can get to know what is the changes happening at each stage. Let's say, for example, someone is going to capture the sample from different states. Let's say these are the states they are going to capture, and they are going to check the expression profile, how the expression profile of many genes is showing variation among these conditions or these time points. So these kind of development study we can do for the with the single cell. We can also go for the cancer research where we can study the tumor condition, finding the microenvironment, finding the cell types for the tumor and expression profile, then try to link with the clinical diagnostics. These things we can do with in the cancer research field. Same thing, we can have the immune response profile, like that we can study how the immune response is behaving against any pathogen or these things. And we can also have the disease mechanism, and also we can have the trajectory analysis. That is basically we can see the development, like how cell is migrating from one state to other state, or how the expression of the gene is showing behavior or like changing conditions. So these things we can also study with the trajectory analysis in a single cell study part. Yeah, now to there are a lot of technology because every year many people are working on these domains, so they're trying to implement new tools, new technology, everything is working. So every year you will get to know, okay, there is a new technology, new company that have discovered new technology to study the single cell data, or let's say spatial data. So this is kind of thing like how the evolution was happening. Like initially it was the people are using the SmartSeq, then CellSeq, Freedom, then Mars, CytoSeq, then 10x Genomics, which is popular. Then also we have SQL. So these are the many platforms which is used to generate the single cell data. And the backend of these platform is basically they use different conditions or different way to capture the single cell and different ways to generate the data. Like the nano droplets in case of some platform. Some platform is using like integrating fluidic circuits, liquid handling robotics part to capture single cell. So there are many different technology in the backend that is used by these kind of platforms. So and we have a lot of platforms that is generating the single cell data. And these are the year when they came up in the picture. Like people are using those platform to do the analysis part. So we have a lot of publications around these platforms. Now after this, we can capture the single cell. So what is the condition we have to do? Because in RNA-seq, we just have to rupture the tissue and isolate all the RNA or all the genome's information, let's say. But when we are dealing with the single cell, so what was the first target? The first target was to isolate single cell. Means we have the tissue, let's say we have this tissue, and the first target is to have the single cell. So I need to isolate all the single cell, and then we can go for the rupturing of the cell to isolate the genomic information. So how we can do that part? So we have a lot of methods. Let's say micro tear plate, microfluidic array, and also microfluidic droplets. There are a couple of methods. Let's say I'm going to explain some basic example concept, how basically we are doing. Okay. So let's say when, so we need to capture the cell also. We need to rupture the cell also, and then we need to do the collection of genomic information, let's say RNA or DNA, and then we need to create multiple sequence, like we need to do the PCR thing to create a copies of those reads. Okay, so we can go for the sequencing. So how we can achieve that? So basically there is a way to capture the thing. So let's say we need to have a droplet, or you can say bubble, while droplet will be there. Okay, we need to fit a couple of things within this droplet or while bubble. So what we have to fit? We need to fit barcode. This bead barcode will be inactive, and then we are going to capture each cell in each droplet. So you can see we have, let's say, 3 droplets. So we are going to place each cell into each droplet. Okay. And the third thing is we need to also add the reverse transcription reagents. Means we have to go for the PCR also. So we need some of the enzyme DNTPs so we can have the chain elongation. So what is happening in one bead? Let's say, for example, so let's say this is a droplet. Now droplet has everything. Like we have a different ways to capture the cells and putting these things all together. Okay. So you can read the backend if you are more interested how the facts flow cytometry to capture the cells is happening, how we use the microfluidic droplets where we will have the barcode, then cells is going to pass, and we will create a bubble or wild droplet where we will have all the inputs that is needed. Let's say beads, cells, and the reagents. Okay. So let's say I'm going to take one example. This one is the one droplet. So if you see the bead barcodes, so remember I was saying when we have the reads, so we are going to add adapter. Okay, so let's say this is the adapter or the tag we add at the end of the reads. Okay, or you can say this is whatever the, you can say fragmented DNA, whatever you want to say, or you can say read. So the general good term is to say the read. So let's say this is the read. So we add the adapter at the end of the reads, and also we add some of the tag. So let me say explain why we are adding a tag. So let's say, for example, in a company, let's say, for example, two person, like for example, there are two brothers who has joined the same company, let's say, for example. Okay. And as of now, let's say, for example, there are 40 employee in a company. Okay. I took the Aadhaar card from all of the joinee, let's say all of the members, and then I have to find like who is siblings. Okay, let's say I have to find who is siblings. Okay, so what are the approach I can do? So one of the approach I can do is, let's say, if I have to find someone like who is siblings, like brothers sisters or brothers, so what are the approach? Because I have the Aadhaar card, so Aadhaar card has the identity for each individual. But for those who are siblings, there is a common thing. Either the address will be common, or let's say the parent's name. So either I can go with the feature to find the siblings using the address, because both are living at the same house. So in Aadhaar card, the address will be the same. So what things I have picked out? Features. So feature is going to say these two person are coming from the same place. Okay. Same thing. Let's say using the same concept, we have a tissue. I have isolated all the cells. Now, or from all the cells, I'm going to isolate the RNA. So how I'm going to say this RNA was captured from this cell, not from this cell. So I need to add a tag to find which RNA was coming from which cell. So that tag is basically the barcode, like Aadhaar card identification. So for each RNA, let's say, uh, instead of using this one, I can use Paint to explain it in a more clear way. So let's say you have cell 1, let's say you have cell 2, and let's say you have cell 3. Okay. Now let's say the RNA I'm going to, so it can have a mixture of things. So let's say, okay, as of now, let's say I'm extracting the transcript for 3 genes. So you can see the color, black is for one gene, blue is for other gene, and this brown is for the third gene. Okay. Now everything is going into a pool, a common pool. Then how you are going to find this particular gene was coming from this particular cell? It is hard to find. So what we are doing before extracting the information or the transcript from the cells, we are adding a tag. So we are going to add, so let's say for this particular tag, this is the tag. So we are going to add a tag to each transcript. So we'll get to know these all transcripts was coming from this cell 1. This is, let's say, this is cell 1, this is cell 2, this is cell 3. So I have added a tag for all the transcript that was coming from cell 1. Same way I can add a tag. So I can add a tag for cell 2. Let's say I have added a tag for the all the transcript that was coming from the cell 2. So let's say the tag was in yellow color. Same thing, I can add a tag in all the transcript that was coming from cell 3. Is it clear?
Yeah, Ashish, we have from a tissue, if we extract cells, we will have millions of cells,
Correct.
right?
Correct.
So, what are these tags made of? Like each cell, if we have to assign one tag,
Correct.
then
It is
it's.
fully a DNA sequence. So it will be a DNA sequence. Let's say some stretch of DNA we are going to create that is going to add to the reads. Let's say this is the reads. With the reads, we are going to ligase this particular tag. So these tag will be unique for each cell. So let's say you have 1K cells. So you are going to have the 1K tag or 1K sequence that you are going to add.
But these tags, since they are comprised of very low nucleotides, right?
Correct.
It's always there are chances that within the transcript, within the reads itself, these nucleotide might be there. So won't it be confusing in those cases? Like,
No, no, no. What we are trying to say, for example, we are adding this tag to do the demultiplexing part. Let's say, for example, now we added the tag for each cell. Let's say I have 3 cells only. Now I have added all the tags. Let's say one tag specific for the cells. So all the transcript that was coming from this cell 1, I have added tag in each transcript. Same thing I did for cell 2, cell 3. Okay. Now I have a pool of things. I'll do all the steps that is common for like elongation, running the piece or everything. So we will have million of reads, let's say. Now what we do, now we do the sequencing. After sequencing, we do the demultiplexing. Demultiplexing means separating the things based on the identity. Now I will have all the reads for cell 1. I will have all the reads for cell 2. I will have all the reads for cell 3. I will have all the reads for cell 4. Okay. Now one thing is sorted, means I have a box where all the transcript was captured for cell 1 is present. I have a box where all the transcript was captured for cell 2. I have a box where all the transcript was captured for cell 3. I have a box where all the transcript was captured for cell 4. Now the second task will come into the role where I have to find the transcript that is present is for which gene. So again, I am going to add one more tag before the experiment, that is going for the UMI. UMI is basically for tagging the feature. Barcode is for tagging the cell. Means barcode will give you the information. This particular transcript is coming from which cell, and UMI is going to give you the information. This particular transcript belongs to which gene. Then only you are going to create the count matrix. Now, like for example, you want to have the count matrix. So what is the number of reads for gene A? Let's
So
say,
basically, when you separate these pools of based on cells or the genes,
hmm,
your coding will include that it should read from the first or the last end where the adapters are usually tagged, right?
correct.
Yeah, because yeah, that's why I got confused that the how unique should be the sequence and millions of sequence like that to be used as tags. So
So
if in between, if these sequences are present and if that is getting caught, then it will be a problem, right? So if the code itself incorporates that we read from the beginning or the 5 prime or the 3 prime end, then that's fine, I guess.
that is taken care at the demultiplexing step, because we are going to read the starting and the last part to read the, because we are aware like we have to read the starting 20 base pair, because I have added a unique barcode that is 20 base pair nucleotide.
Mm-hmm.
So I'm going to check which
Right.
one is matching, and then I can pick it. Like I need to pick a black one with the blue tag to this box. I need to pick black one with yellow tag to this box. So this is the part of demultiplexing process.
Hmm.
So now up to this part, we only have the information like which transcript is coming from which cell.
Correct.
But you are also aware that within a cell, we have a transcript that belongs to different gene.
Correct. Yeah, yeah.
So for
Hmm.
that identification, now I need to identify at the gene level. We were doing the identification or demultiplexing at a cell level.
Hmm.
Now at a gene level, again we have a one tag. So I guess I may have the image. Do I have? Okay, I can quickly search for NX Genomics barcode and EMI, let's say. Yeah, on this one. So let's say this is your region of interest. So hmm. So the I was saying we have a bead barcode. Okay. So the bead barcode will have a couple of things, and that part we are going to add a lot of information, like let's say barcode and UMI. So barcode is basically for identification of cell, like which transcript is coming from which cell, and UMI is basically this is unique for each gene, this is unique for each cell. So these two concepts are there. Barcode, when you are like whenever you are going to hurt barcode, it means this is unique for each cell. So we need to have the barcode for demultiplexing to collect which cell is like which transcript is coming from which cell. UMI is basically for like we may have multiple transcript for the same gene. So for all the similar genes, let's say gene A has 5 transcripts, so all the 5 transcripts will have the same UMI. Then we will have gene B. Let's say gene B will have 3 transcripts. So all the 3 transcripts will have different unique UMI. Okay, so this is the way we are going to capture and do the demultiplexing part. Because the target is this one. I need to have all the million, billions, whatever the cells we have within this tissue. We are going to have those information, and we have to know the count or the number of transcript that is present for each gene. So within a cell, we collected all the transcript that is coming from cell 1. But within those transcripts, you should also know which transcript is coming from which gene. So that part is coming from UMI, and which transcript is coming from which cell is coming from the barcode information. So these are the two tags that is helpful to get these two information. Yeah, so I was explaining the concept on the paint box. So yeah, this is the inactive bead, and within the inactive bead, we are going to attach some reads, and this read will consist of couple of things. Let's say cell barcode, UMI, oligo DTs. Okay, because and what then the process will happen. Let's say in the general, if I say you have a bead and you have a lot of sequence, that will be the complementary of the adapter you are going to add. Let's say this is your read, okay, and both the end of the reads you are going to add the adapter, and whatever the adapter sequence, we are going to have the complementary sequence in this particular thing. So this is going to bind in this way. Okay, so let's say this is your thing, so this is going to bind, this is going to bind, so you will have on the beats couple of reads is going to bind with the complementary adapter sequence. And then we can have the natural process like the chain elongation, adding the nucleotide base pair, and after creating the entire stretch, we can denature it and it will separate out. Then again the same process will repeat. Again the bead will be free to take more transcript, do the sequencing, create a entire elongation of the chain, separate out. So this is the process. So there will be a many beads and within the things, and then they are going to capture the information. So how we basically capture and how we can have the information which transcript is coming from which cell, and how we can get the number of count of transcript for each gene. So get the count for each gene, we use the UMI, and cell barcode is basically to give you the identity which transcript is coming from which cell. So after covering this topic, now if you see the entire whatever the things I was covering is that basically the part of the wet lab part, or the to understand the how people used to prepare the library, what are the information we should know about those things. And after generating the data, because after having all the sequencing reads, we will have a million of copies, and we are going to send it for the process third that we were discussing in the first slide, that is for sequencing. So we are going to send it for the sequencing for our samples, and sequencer will do the sequencing, and then we will get the raw data, that is the FASTQ file. And from the FASTQ file we will have a couple of workflow. Okay, so this is basically the entire or the typical single cell RNA-seq workflow. So basically we are going to start with the raw data. So raw data is basically your FASTQ. Okay, and from the FASTQ you need to do the alignment and need to generate the count matrix. So you will have the count matrix. So what is the count matrix? This is the count matrix. Count matrix is basically the 2D matrix. 2D matrix in the sense you will have the information, rows will have the all the genes that is present within the sample, column will all the cells that is part of the tissue. Okay, so the tissue may have 1k cells, 2k cells, whatever the cells you are going to capture, you will have those information in the column part. And how we are going to read the count matrix, you're going to say the number of reads, or you can say number of transcript, whatever you want to say. Let's say I'm going to say the number of reads that is mapping to gene 1 is 3, and they are present within the cell 1. So how many reads for gene 1 present within the cell 2, how many reads that is present within the cell n for the gene 1. So this is basically the count. Count means the expression profile. So what we can conclude from this, you can simply say this cell number or whatever the cell this is showing high expression profile for the gene 1 compared with the cell 1 and cell 2. You can see the expression profile is 13, means there are 13 transcript that is present for this particular gene in this particular cell. So these things we can read it from the count matrix. So we will have the count matrix, and from the count matrix, what we have to do, we have to do the quality control. Because every time whenever you are going to start, so quality control steps is very important, because a lot of complication or a lot of thing is happening in the wet lab part. So you need to check the quality of the data. So you need to do the quality control while doing the alignment to check the quality of the reads, whether the sequencing was happened correctly or not, whether we have to redo the sequencing or not. Those information we need to check. And also at the cell level, because it's not every time, whenever you're going to create a bubble, it's not a case like every time you're going to capture one cell. Sometimes there will be a while bubble, or you can say droplet, where you will have the beads and other information and cell is missing. Sometimes you will have the droplet where you will have more than one cell. This is the case of doublet where we have two cells captured. So this is not giving you the single cell expression profile. Now whatever the information you are going to get, that will be double case, like whatever the transcript that is coming from this one and this one, they are going to combine and give you the expression profile. So this is the bias thing. So this will create the bias in the downstream analysis, because this is wrong, because we were thinking all the cells having the gene expression profile at a single cell level, but when you are comparing this cell with this cell level, so this is not correct, because this already have two cell combinations, so obviously this will have a higher transcript level. So every time it will say this is showing high expression profile. But if you see the backend, the problem was because this particular droplet was capturing two cells. So that's why it has more number of transcript compared with this particular set. So these things comes under the quality control part, that we have to do and take care of that part in the quality control step. So we have a lot of quality control step with the raw data, that is the count matrix. This is the first step. Then we have to go for the normalization. Why normalization? Because comparing between different samples, we need to have the normalization. Because let's say for example, if I have to compare between person A and person B, let's say person A has 5 apple and person B has a 10 apple. So we can't do the comparison because there is a no similar category thing. So what normalization basically do? It will take 5 apple from this part and randomly it is going to pick 5 apple from the 10 apples to do the comparison, like whoever having the good quality of apple, let's say I want to do the comparison, let's say. So this is just a concept of normalization. So we used to normalize the data in a such a way to make the sample comparable, so we can do the comparative analysis. Okay, so if we have to compare between cell 1 versus cell 2, so we have to do some normalization. Okay, let's say if we have to compare between, let's say, normal sample versus tumor sample, so normal sample will have, let's say for example, 1k cells and tumor cells may have a 2k cells. So we have to do some normalization to do the comparison between normal versus tumor. So that's the way we use the normalization to handle those kind of situation. Now batch effect correction. Because why we need to do batch effect correction? Let's say there's one good example. I may have slide now. Okay. So let's say batch effect correction. Simple example. Let's say there is a three students. Let's say A, B, and C. Okay. If I am going to give a task to each one of you to measure the height of a tree, okay, so everyone is going to measure some different height, okay, because there is some variation or manual error part that comes under. So basically, for example, if a person is going to measure the height, let's say 200 centimeter, 200 meter, someone is going to measure the height to 10 meter, and C person is going to measure the height at 180 or 185. So there is a difference in the results that we are going to see in the downstream process. And also, let's say, for example, if all the three person, I'm going to ask, could you please repeat the single cell wet lab experiment? I'm going to provide everything, the same thing, the solution, buffer, whatever the things that is required to perform the single cell experiment, but you will find the difference in the results. Why? Because the handling part, someone is good at handling the pipetting, someone is not too good at handling the pipetting, someone is not good at handling and storing the solutions and buffer and everything. So there will be variation in the results in the output part. So I don't want those variation will hamper my result or create bias. So those things is basically captured or resolved in the this particular part, that is batch correction. Before batch correction, there might be variation, but when we are going to taken care of these things, then we are going to have the good results that will be comparable. There will be no bias. So that time we have to do the batch correction when we are going to handle multiple samples. Okay. And after having the batch correction, we can go for the downstream analysis, basically the clustering part, because the main purpose was to find the cell types or find the heterogeneity of the tissue. So let's say what are the cell types, or what my entire tissue or my sample consist of which kind of cells. So this is the part of cell annotation. After getting the number of clusters, each clusters we are going to annotate one cell type. So this comes under cluster annotation, or you can say cell type annotation. And after cell type annotation, basically we are going to follow the other downstream process that is important. First is differential expression gene analysis. You might have seen in RNA-seq also, where we are going to find the genes that is showing high expression in one condition compared with the other condition. Let's say we have a sample tumor versus non-tumor for the single cell study. We can find the markers or expression profile between these two conditions, and we can have a list of upregulated and downregulated genes using D genes analysis. Same way, we can have the trajectory inference that I was explaining the role of trajectory analysis. If you want to see the expression profile that is changing between different cell types, like we are doing some development studies, so how a cell is migrating from one cell state to other cell state, that we can do using the trajectory analysis. And also we can have a couple of part like gene dynamics we can check. We can have the composition analysis and other pathway analysis, network analysis, a lot of things people used to do depending upon the questions they are trying to solve using their experiments. Okay, but this is the basic structure of single cell analysis workflow. After this, yeah, so I was saying when we have the sequencer, so either you can get the output as the FASTQ, or before FASTQ, some is like sequencer generate the BCL file format. Illumina basically generate the intermediate file format. This is binary base call file, and this will store all the information that is present within the FASTQ file. The only difference is this is not readable, so we need to convert BCL to the FASTQC file. Okay, so FASTQ file, how we are going to convert, because Illumina has already an open tool that is BCL to FASTQ conversion. You can use it to convert and you will get the FASTQ file. After FASTQ file, I was explaining, we have to do the alignment, we need to do some correction, and we need to reach a stage that is the count matrix, generation of count matrix. A count matrix will have the information for the gene, cell, and their count. Means the number of reads that is present for gene 1 within a cell 1, number of reads for gene 1 for cell 2. So these are the count matrix. So we need to reach to this stage. So let's say if you're doing your experiment, so you will face these things, like starting with the FASTQ file, doing the alignment and everything. Let's say you're going to use some database to download some single cell data. So you will start from this one, because mostly some of the database you're going to use, they basically try to upload the count matrix. So anyone can use the data to do some validation and do some findings. Okay. So now the important point is how you are going to find the data. So if you're aware of database, so we have a lot of database for a single cell. Let's say NCBI. You can also go for the 10x, 10x platform. There you will find many single cell data. Single cell portal is also there. So I may have, yeah. So this is the single cell portal. You simply have to search single cell portal, and when you click on single cell portal, you will have a lot of information of all the single cell experiments. You can do the filtering part, you can select different conditions. Let's say for example I want to have the particular cell type of particular tissue, so you can just add in the search box and you will get all the single cell experiment related to that particular tissue. Let's say for example I am interested for the mouse brain data. If I'm going to write mouse brain, then we can have a list of all the single cell experiment that was uploaded to this database related to single cell. Okay, so this is the way to collect the information. Or either you can go to NCBI. NCBI has also a lot of database. gene expression omnibus, if you heard that particular term. Yeah, this is the part. Now let's say 10x platform has also generating the data. So there are two ways. You can simply follow the normal alignment, or for example, you don't want to follow, because if you're going for the open source tools, that time you need to use a couple of tools for doing a different task. Let's say for alignment, you're going to use HiSet or maybe the star aligner. For example, for the generation of the count matrix, you're going to use feature count. Cell Ranger has compiled those things. You just need to know how to use this particular tool. This will take your input file FASTQ files, and in the output files they are going to generate the count matrix. Okay, so this is taking care of all the things. You don't have to, you just have to explore the cell ranger. Whatever the parameters that is needed to use, you need to explore those parameters, how to tweak those parameters, how to modify the parameters to generate a good alignment results. And then in the output files, you will have a couple of output files, but we are only going to focus on these two. So these are the two files you can see. Like, this is I have taken from the Cell Ranger documentation itself. So it will have a couple of files. I'm going to focus on these two files, highlighted blue one, raw feature barcode matrix and filter feature barcode matrix. So what exactly these two files are? So basically these two files are the count matrix file. Count matrix in the cells, this file. Okay. But these files will have this structure as of now. They are going to provide the structure in this kind of thing. Means there will be three files. You need to use these three files to combine and create a 2D structure count matrix. So what are those files will be present within these two structure? Barcodes file. Barcode file will have the unique barcodes of each cell. Feature file will have all the information for the features that is captured from the cell. Matrix file will have the count information of the unique EMI for each gene in each cell. So these three files you need to combine to create a 2D matrix. This way, like you have 2D matrix. So there are lots of package function is available. You just need to use it to convert it. Okay. Now coming back to this part, what is the difference between raw and filter? Because sometimes if you're going to download the data from 10x, you will encounter with these two files. Okay, so both the files are the compressed version .gz. If you're going to uncompress it, then you will get these three files. Okay, so raw and feature, what is the difference? Because the 10x also do some pre-processing. As I was saying, we have to do the quality control to filter out poor quality cells. So there will be no bias in the downstream analysis part. So this is the raw data which will have all the cells, and this is the filter data. Means the 10x has already performed some of the filtering condition or some of the quality control. So let's say for example this feature barcode matrix may have, let's say, 5k cells. So after applying some filter parameter, this may have, let's say, 3.5k. Let's say this may have 3.5k. Okay, so it's up to you which file you want to use. You want to start from the raw scratch file and you want to apply your filter, or let's say you already want to use some filter data and you want to apply some other filter parameter. So you can apply both the condition. It's totally up to you how you want to proceed. Okay, so I just explained these two file format you will encounter when you're going to deal with 10x data. Okay, and then you're going to extract. So these are the file format. If you're going to download the data from some other database, let's say NCBI or let's say a single broad thing, so there might be you're going to have a simply count matrix. You don't have to do the combination. There you will have a count matrix.gz file that is already converted these things into this one. So you already have the count matrix. Okay. So whenever you are going to use NCBI or the single cell portal where you are going to find the count matrix file directly, when you're going to deal with 10x platform to download the data, you're going to encounter with these two files, and when you're going to unzip the files, you're going to get these three file formats, and you need to read these file format to convert into a current matrix or the 2D matrix files. Yeah, now coming to the analysis part. Like as of now, we get to know what is the backend, how people do single cell experiment, what are the applications, and what is the general workflow. So to do the analysis part, we need to know the coding language to write the codes. So there are two platforms. Either someone can do the entire analysis in R based platform, or the second is the Python based. So for the R base, there is a already package that we call Surath. So Surath has all the inbuilt method they have created already. You can simply use those method to do the analysis part. You can also create your method if you are too interested. If you are not interested to create your method or you don't want to spend much time, so you can use the existing method from the Surath package. The second one is Python or ScanPy. Okay, so let's say for example, if I'm going to showcase Surath one, so you need to go to this particular thing, and there is a get started thing. Here they have provided tutorials for all of different kind of analysis. Okay, like single cell, other kind of integration method. Then we have the spatial thing. So you can explore. Let's say I'm interested for the single cell. So this is the entire analysis for handling the single cell data, and the entire script or the code is basically R based. Okay. So to get to know or to understand what is happening with this code, you should know the R programming. This is the first condition. And then you can try to execute the code with your data, or you can also try with the data they are using, like they are using the PVMC data. So you can generate different kind of plots and then from these plots you have to conclude to what are the filtering parameters I need to use. So these are basically the QC plots. So I'll explain these plots in maybe upcoming session if I'm going to take. So that time you can, or even you can explore by your own, like what exactly these plots and how we can conclude. So what are these plots, how these plots are helpful for the quality controls. So this is the part of Sura that is R based. Then you have one more package. This is the SquidPy. SquidPy is basically handling for the single cell and the spatial also, and this is based on Python. So let's say someone is too comfortable in Python, they can go for the SquidPy, and someone is too comfortable with R, they can go with the Surath package. It's totally up to you on which platform you are more comfortable. But also there are a lot of advantage and disadvantage because there are many features which are present in R might not be able in present in the Python. There are many modules or package that is present in Python might not be present in this one. So people used to do both the things. Let's say some of the features they try to cover with this one. Let's say I need to generate some plots from the package that is present in Python. So I can do the same thing with Python. Okay, so this is the two platform we can use to analyze the data. And the last part is now what domain knowledge we should have to analyze any single cell data. So first concept is we should know the single cell. So like what exactly the single cell means. So what is the experimental approach? So what are the techniques and methods? What is the barcode UMI? What is demultiplexing? What is coverage? So these things you should know before jumping into any other domains. So what is the structure of input files? What is count matrix? What is expression profile? What is gene expression data? So these things should come under single cell domain. So you should read about single cell. Second thing is Linux command line because you are going to play with the data, you are going to write something. So it's good, for example, whenever you're going to download the data, so the data is will be in the compressed version. So let's say for example, if you have to uncompress it, you can use the command line. If you're comfortable with Indo, you need to use some other application that is going to do the decompression part for you. Then the third domain you should know is one coding language, R and Python. See, both the coding language is easy. Let's say if you are good in Python or if you're good in Python R, so you can switch between different coding language. The difference is only the syntax part. The rest of the concept, logic, is everything same. How to define a variable, how to run a loop, how to call, how to load the package. Everything is very simple. If you're comfortable in writing and applying the logic in one coding language, you can simply do in other coding language. That's very easy. Third part is the package and library installation. How to deal with package, how to deal with library, how to install those packages so I can use the method that is already defined. So if you are aware of Python or R, so there is a package or modules we say that is already developed by someone else. I just need to use the method. I need to call the method and use it for my data to do the analysis part. So those comes under the package and library installation. Next one is the database, because for the single cell data, I should know from where I can download the data. So you should aware of the database like NCBI, single cell portal, and also gene expression omnibus, and also some of the database for annotating the cell type. Because to annotate the cell type, we need the database of marker genes, or you can say cell types, that we are going to use it as a reference. And at the last, data visualization means you should know how to present the data. Generating the result is very easy. You can copy the codes. You can copy the codes from any of the resources, because there are a couple of resources present on YouTube, GitHub, on single cell. You can copy the code, analyze the data, but it's hard to present the data in a good visualization mode. So you have to work on how you can showcase your data and how you can conclude something meaningful from the data. Yeah, so this is the entire session for the single cell part. Thank you. And yeah, please make sure to like and subscribe and share the video.